10 questions, cardiac muscle and cardiac output. Pause after each question if you need time to think. Let's go. Question 1. The contractile unit of cardiac muscle is the A. T-tubule, B. Intercalated disc, C. Sarcomere, D. Gap junction. Pause if you need to think. The answer is C. Sarcomere. Here's why. The sarcomere is the fundamental contractile unit of cardiac muscle and skeletal muscle. It extends from Z-line to Z-line. Within each sarcomere, thick filaments of myosin and thin filaments of actin are precisely arranged in overlapping arrays. When calcium binds troponin C, the troponin-tropomyosin complex shifts position, exposing the myosin binding sites on actin. Myosin heads bind actin. The power stroke occurs. The Z-lines are pulled toward each other. The sarcomere shortens. Millions of sarcomeres shortening simultaneously, in series and in parallel, produce the coordinated contraction of the ventricular wall, generating the pressure that ejects blood into the aorta. Every other structure on this question, T-tubules, intercalated discs, gap junctions, plays a supporting role in getting the signal to the sarcomere, or connecting sarcomeres together. But the sarcomere itself, actin, myosin, Z-lines, is where contraction actually happens. Board Pearl. Sarcomere equals Z-line to Z-line. Fundamental contractile unit. Cardiac and skeletal muscle. Question 2. Which structure maintains cell-to-cell cohesion in cardiac muscle? A. Sarcomere, B. Intercalated disc, C. T-tubule, D. Sarcoplasmic reticulum. The answer is B. Intercalated disc. Here's why. Intercalated discs are specialized junctional complexes that connect neighboring cardiac myocytes end to end. They contain two critical components: mechanical junctions, desmosomes and fascia adherens. These physically anchor adjacent myocytes together, transferring the mechanical force of contraction from cell to cell without tearing the myocardium apart. Think about what happens during ventricular contraction. Enormous mechanical forces are generated. Individual myocytes are shortening forcefully. Without strong cell-to-cell connections, the cells would simply pull away from each other. Intercalated discs prevent this. They are the mechanical glue of the cardiac syncytium. Electrical junctions, gap junctions, also located within intercalated discs, allow direct electrical communication between adjacent cells. Ion current flows from cell to cell. The action potential propagates rapidly through the myocardium, allowing coordinated contraction. Mechanical cohesion and electrical coupling, both housed in the intercalated disc. This dual function makes the intercalated disc one of the most elegant structures in cardiovascular physiology. Question 3. The primary calcium release channel of the sarcoplasmic reticulum is the A. SERCA pump, B. Sodium-calcium exchanger, C. Ryanodine receptor, D. Dihydropyridine receptor. The answer is C. Ryanodine receptor. Here's why. This is the heart of excitation-contraction coupling, the process that links the electrical action potential to mechanical contraction. Here is the sequence. The action potential depolarizes the cell membrane, travels down the T-tubules, reaches the dihydropyridine receptors, voltage-sensitive L-type calcium channels on the T-tubule membrane. These open. A small amount of calcium enters the cell from outside. That small calcium influx triggers the ryanodine receptors, calcium release channels, on the sarcoplasmic reticulum membrane. The ryanodine receptors open, and the SR releases a large flood of stored calcium into the cytoplasm. This is calcium-induced calcium release, a small trigger producing a large release. The released calcium binds troponin C. Contraction occurs. After contraction, the SERCA pump, SR calcium ATPase, actively pumps calcium back into the SR, restoring the calcium store, allowing relaxation. Ryanodine receptor, release. SERCA pump, reuptake. Two sides of the same cycle. Board pearl. Ryanodine receptors are the calcium release channel of the SR. SERCA pumps calcium back in. Dihydropyridine receptors are the trigger on the T tubule. Know all three and their roles. Question 4. Digitalis increases contractility by A. Activating beta-1 receptors, B. Blocking L-type calcium channels, C. Increasing potassium efflux from myocardial cells, D. Inhibiting sodium-potassium ATPase. The answer is D. Inhibiting sodium-potassium ATPase. Here's why. The mechanism of digoxin is one of the most elegant and testable pathways in pharmacology. Follow the chain. Digoxin inhibits the sodium-potassium ATPase pump, the pump that normally moves sodium out of the cell and potassium into the cell. When this pump is inhibited, sodium accumulates inside the cell. Intracellular sodium rises. The sodium-calcium exchanger, NCX, normally exports calcium from the cell in exchange for importing sodium. It relies on the sodium gradient, low sodium inside the cell, to drive calcium out. When intracellular sodium rises, that gradient is reduced. The sodium-calcium exchanger cannot remove calcium as effectively. Intracellular calcium rises. More calcium available for excitation-contraction coupling. Stronger myocardial contraction, positive inotropy. The complete board chain. Digoxin inhibits Na/KATPase. Intracellular sodium rises. Sodium-calcium exchanger activity decreases. Intracellular calcium rises. Stronger contraction. This is also why hypokalemia potentiates digoxin toxicity. Hypokalemia means less potassium competing with digoxin at the Na/KATPase binding site. Digoxin effect is amplified. Toxicity risk increases. Check potassium before and during digoxin therapy. It is not optional. Question 5. Which structure stores calcium for excitation-contraction coupling? A. Sarcoplasmic reticulum. B. Mitochondria. C. Intercalated disc. D. T-tubule. The answer is A. Sarcoplasmic reticulum. Here's why. The sarcoplasmic reticulum, SR, is the major intracellular calcium reservoir in cardiac muscle. It is a specialized network of membrane-enclosed tubules that wraps around the myofibrils in close proximity to the sarcomeres. This proximity is essential. Calcium released from the SR only has to travel a very short distance to reach troponin C and initiate contraction. At rest, the SR actively maintains high concentrations of calcium in its lumen, primed and ready for release. This calcium is sequestered by calsequestrin, a calcium-binding protein inside the SR that allows the SR to store far more calcium than would otherwise be possible. Upon stimulation, ryanodine receptors open, calcium floods out, contraction occurs. Then SERCA pumps, powered by ATP, rapidly return calcium to the SR. The cell relaxes, and the SR is reloaded for the next beat. This cycle, store, release, reuptake, reload, occurs with every single heartbeat. Clinically, heart failure is partly characterized by dysfunction of this cycle, reduced SR calcium stores, impaired ryanodine receptor function, weaker contraction, decreased contractility. Many inotropic agents, both pharmacologic and physiologic, work by modifying this calcium cycling process. Question 6. Which of the following best explains why cardiac muscle cannot undergo tetanic contraction? A. Lack of calcium channels. B. Presence of gap junctions. C. Long absolute refractory period. D. High mitochondrial content. The answer is C. long absolute refractory period. Here's why. In skeletal muscle, repeated rapid stimuli can summate. The muscle stays in a continuous contracted state, tetany. This is useful for sustained voluntary movement. In cardiac muscle, tetany would be fatal. A heart that cannot relax cannot fill with blood. No filling, no stroke volume, no cardiac output. The cardiac action potential prevents this with elegant simplicity. The plateau phase, phase 2, is prolonged, lasting approximately 200 to 250 milliseconds. During this entire plateau, the cardiac cell is absolutely refractory. Absolutely refractory means no matter how strong the stimulus, the cell cannot be re-excited. The sodium channels are inactivated. They are not available for another action potential until repolarization is nearly complete. By the time the absolute refractory period ends, the cell has already repolarized. Mechanical contraction has ended, and the heart is ready to relax. The absolute refractory period is almost as long as the contraction itself. This timing is not accidental. It is physiologically essential. It ensures that each heartbeat is followed by relaxation, diastolic filling, before the next beat can occur. It is the built-in protection against tetanic cardiac contraction. And it is what makes the heart fundamentally different from skeletal muscle. Question 7. Which protein binds calcium to initiate contraction? A. Tropomyosin. B. Myosin. C. Actin. D. Troponin. C. The answer is D. Troponin C. Here's why. The troponin complex sits on the thin filament alongside actin. It has 3 components. Troponin C, the calcium-sensing subunit. This is the one that matters for contraction initiation. Troponin 1, the inhibitory subunit. At rest, in the absence of calcium, Troponin 1 holds tropomyosin in a position that physically blocks the myosin binding sites on actin. Troponin T, the tropomyosin binding subunit, anchors the complex to tropomyosin. Here is what happens when calcium arrives. Calcium binds Troponin C. This binding changes the configuration of the entire troponin-tropomyosin complex. Tropomyosin shifts position, moves away from the myosin binding sites, exposing them on actin. Myosin heads can now bind to actin. Cross-bridge cycling begins. The power stroke occurs. Sarcomere shortens. Contraction. When calcium is removed, reuptaken by circa, Troponin C releases calcium. Troponin 1 re-inhibits. Tropomyosin covers the binding sites again. Contraction stops, relaxation occurs. Clinically, Troponin 1 and Troponin T are the biomarkers measured in acute myocardial infarction. When myocytes are damaged, these proteins leak into the bloodstream. You are measuring the structural proteins of the contractile apparatus itself. Troponin C, calcium sensor, initiates contraction. Troponin 1 and T, markers of myocardial injury. Know the distinction. Question. The major energy source used by the myocardium to generate stroke work is A. Glucose. B. Lactate. C. Amino acids. D. Fatty acids. The answer is D. Fatty acids. Here's why. The heart is an aerobic organ. It almost never rests. It cannot sustain oxygen debt the way skeletal muscle can. It requires continuous ATP production. Under normal fasting conditions, fatty acids provide approximately 60 to 70% of myocardial ATP. The heart oxidizes fatty acids through beta oxidation, generating acetyl-CoA, which enters the Krebs cycle, producing large amounts of ATP through oxidative phosphorylation. The heart is extraordinarily efficient at extracting ATP from fat, which makes sense. Fat provides more ATP per gram than any other substrate. But this preference shifts with physiologic state. After a carbohydrate-rich meal, glucose utilization increases. During intense exercise, lactate becomes an important substrate. The heart actually consumes lactate produced by working skeletal muscle. During ischemia, fatty acid oxidation is impaired because it requires oxygen. The heart shifts toward anaerobic glycolysis. ATP production falls dramatically, which is why ischemia so rapidly compromises contractility. This metabolic flexibility is one of the heart's remarkable adaptive features. But the baseline preference, the fuel the heart uses during normal resting conditions, is fatty acids. Board pearl, fatty acids, 60 to 70% of myocardial energy at rest. Glucose increases post-meal. Lactate during exercise. Anaerobic glycolysis during ischemia. Question 9. Myocardial oxygen supply is most dependent on A. Coronary blood flow, B. Heart rate, C. Contractility, D. Stroke volume. The answer is A. Coronary blood flow. Here's why. The myocardium is unique in its relationship with oxygen. Most tissues in the body extract approximately 25% of the oxygen delivered to them, leaving a large venous reserve. The myocardium, at rest, already extracts approximately 70 to 80% of the oxygen delivered by coronary blood flow. This is near maximal extraction. There is almost no reserve left to tap. In most organs, when oxygen demand increases, the organ can simply extract more oxygen from the same blood flow. The heart cannot do this to any significant degree. It is already extracting almost everything available. So when myocardial oxygen demand increases, the only way to meet that demand is to increase coronary blood flow. More flow, not more extraction. Flow is the primary determinant of myocardial oxygen supply. This is why coronary artery disease is so dangerous. A fixed stenosis limits the ability to increase flow, even when demand increases with exercise, or tachycardia, or increased afterload. Supply cannot match demand. Ischemia occurs. And this is why the management of myocardial oxygen balance in anesthesia focuses on maintaining coronary perfusion pressure, avoiding excessive tachycardia, avoiding dramatic increases in wall stress and afterload, all designed to keep supply in excess of demand in a heart that has almost no extraction reserve. Coronary blood flow is myocardial oxygen supply. They are essentially the same thing. Question 10. Which factor primarily determines ventricular filling during late diastole? A. Atrial contraction. B. Aortic pressure. C. Ventricular systole. D. Pulmonary vascular resistance. The answer is A. Atrial contraction. Here's why. Ventricular filling during diastole occurs in two phases. Early diastole, passive filling. The mitral valve opens. Blood flows from the left atrium into the left ventricle passively, down the pressure gradient. No atrial contraction needed. Approximately 70 to 80% of ventricular filling occurs passively in early diastole at normal heart rates. Late diastole, active filling. Atrial contraction occurs. The P wave on the ECG. The atrium squeezes, actively pushing the remaining volume into the ventricle. This atrial contribution, the atrial kick, contributes approximately 20 to 30% of total ventricular end diastolic volume at normal heart rates. This may seem modest, but consider when it matters most. In a patient with diastolic dysfunction, a stiff, non-compliant ventricle, passive filling is impaired. The stiff ventricle resists filling. The atrial kick becomes disproportionately important, sometimes contributing 40% or more of total ventricular filling. This is why atrial fibrillation is so poorly tolerated in patients with diastolic dysfunction, hypertrophic cardiomyopathy, aortic stenosis, hypertensive heart disease. Loss of the atrial kick in a heart that is already dependent on it can precipitate acute decompensation and hemodynamic collapse. In anesthesia, This is why restoring sinus rhythm, or controlling rate to allow adequate diastolic filling time, is critical in these patients. The atrial kick is not optional for a stiff heart. That is 10 questions. Cardiac muscle and cardiac output. I'm Anesthesia Buddy. Stay sharp. See you in the next one.